Okay.
All right. So thanks for coming. My name is Damon Ty. I'm Bio-Rad's curriculum and training specialist from the Western Coast. Together, I've got Lee Brown here and Tamika Stubbs, who did a bunch of the heavy lifting for this webinar, in that they've made your plates, got you worms, and got you all set up to be able to do this hands-on. And I'm getting the easy job today of just talking you through it. So with that, let's go ahead and kind of drop into today's webinar. So we're going to be working with C. elegans. We'll do a little introduction about C. elegans. How many of you here have actually worked with them before? Go ahead and put that in the chat if you've worked with C. elegans before. It's a model organism that is very, very widely used in research, but I find that it's not touched a whole bunch, at least at the high school level, and even still a very, very low amounts in the college level as well. Oh, okay. So a handful of you have. That's great. So those of you who have worked with them before, you know that they're a pretty easy organism to work with, as far as easy to culture and things like that. So that's some C. elegans underneath a dissecting scope that one of our R&D people shot while trying to build this lab years ago. The beauty about C. elegans is that they're a multicellular organism that's really easy to keep, especially compared to a lot of the other multicellular organisms you might use as a model organism. They've got a fully functioning nervous system, and this is probably where I say they're kind of leading the pack, is in the ability for us to model connectomes or nervous systems at a grand scale within them. Because I mean, you can see them, you can basically look right through them and see the nervous system. It's been well mapped, and we'll dig a little bit more into that later. They've also got a great number of genetic tools that have been developed, full genome sequencing. We know the fate of every single cell of the C. elegans. So from when you have a fertilized egg all the way to when it gets this 939 celled adult, we know what every cell does. When it divides, where's it going to go, all that stuff. And that was all mapped out using something a lot of you guys are probably familiar with, GFP. They're super easy to culture. So if you've ever been able to culture E. coli in your classroom, you'll be able to culture C. elegans. Mainly because we're going to feed them on E. coli to make them really easy to work with. And then, like I said before, a super widely used model organism, and which is amazing because they haven't been around that long as a model organism compared to a lot of the other model organisms you're probably aware of. And it was mainly brought in by Sidney Brenner, who's kind of a very kind of visionary person within biology, and he saw the power of having a multicellular organism that you could easily grow and that you could see through. So the workflow today is we're actually going to get started right away with doing a lab protocol, which is unusual. Usually it's good to dive into the theory first so you know what you're doing. But the reason we're going to start with a lab protocol today is because the chemotaxis that you're going to be performing takes about 30 minutes for the worms to decide where they're going so that we can analyze the results. And so we need to kind of just get that started so that way that can happen in the background, and then we'll step back and do a bunch of the theory while those worms are moving their way around their plate and deciding where to go based upon their ability to learn or not learn. You should have received lab stations in the mail. This is the lab stations that you would normally see at a student workbench. The differences today is that most of you probably do not have a P20 micropipette and tips laying around, which is fine. We're going to be able to do everything today with just the transfer pipettes that you received in your packages. These other things are more essential. So you should have an NGM plate with wild type worms, an NGM plate with mutant worms, and then assay plates that you would have added salt and water to last night to diffuse. You should have some wash buffer, empty 1.5 mil tubes, those transfer pipettes. And if you've got a smartphone around, bust it out. Smartphones make it a lot easier to see the worms in two regards. Some of your smartphones now have like macro lenses built into them, and you can just zoom in and be able to see the worms. Or if you're not going to look that close, you turn on your light and you put it on the side of the plate, so you got basically light coming at a 90-degree angle to the worms, it makes it a lot easier. A higher contrast will happen on the edge of the worm, and you can kind of see them with the naked eye. A Sharpie will also help because we're going to be labeling some of your tubes and your transfer pipettes. And then, really importantly, find yourself a waste cup, because you will have waste liquids with E. coli in them. And then if you've got a little piece of like paper towel around, that might be helpful if you end up with any spills or a lot of water on your plates. Does everybody have all those items? Everybody's good? If you don't have that, or you've got a question about the lab station materials that you have, just let us know in the chat, and Tamika or Lee could kind of step in and help with that. So first things first, we gotta understand the primary liquid handling device we're using today, which is a transfer pipette. And sometimes as a teacher, I would forget to show the students how to use this, and it would always come back to bite me in the butt. And so it's always really important to show them, this is your transfer pipette, this is how you use it. Here are the gradations of the pipette and what those volumes are. So predominantly today, we're going to be using the lower volumes of the transfer pipette, so 500 microliters kind of down to, actually going to go beneath 100 microliters today for some of this. The big thing, though, is, of course, to demonstrate to students that the way to use one is you squeeze first, go into the liquid, gently release until you get to the level, the volume you want, and then remove yourself from the liquid and then go to where you want to deposit it and gently squeeze again. If you don't do that, you of course know what students are doing, right? They're putting the transfer pipette into the liquid and then squeezing as fast as they can, and stuff's going all over the place. So it's good just to give them that reminder before getting started. So let's pull out your plates and some of the flip-top tubes and your Sharpie.
So, Damon, I was going to mention, so I've got my camera on here. I think it's spotlighted for everyone. What that means is that if you go to view options, you should be able to click on side-by-side mode. And then you can look at the PowerPoint and this screen, the video right here where my hands will be side by side. You can drag the slider in the middle and make it bigger or smaller. So that's what's going on there. So I'll be doing the lab with you guys.
Lee, if you would take a moment and repeat that slowly one more time, just to make sure everyone is on board.
Absolutely. So up at the top of your screen where it says you are viewing Damon Ty's screen, there's something called view options. And if you click on that, there is an option to do side-by-side mode. And what that'll do is it will open up both my camera and the PowerPoint together. And you can drag the slider one way or another to see the, my picture bigger or smaller.
Very good. And so Lee's going to be demonstrating the same movements you're going to be doing at home with your materials, so that way you've got a visual read of how to do this, because words are great, still pictures are good, but actually being able to see it live is even better. So the first thing we're going to have you do is find two 1.5 mil tubes, and I'm going to have you label them with your Sharpie. WT on one of them for wild type and M for a mutant on the other one. So give you a couple minutes or a minute to do that. And then once you've labeled those, grab your two assay plates that you set up last night and label one wild type and one mutant. I usually label it on the bottom side of the plate, so on the auger bottoms at the rim. And the reason I usually label it right at the rim is just because if I'm going to be trying to count something later on, I don't want a giant bunch of letters in the center while I'll be trying to count against. And then once you've got those labeled, we can move on to the next step. And I'm going to talk you through the next step before having you jump in, just because it's a little bit, it's not super involved, but just good to hear it all the way through so you have a visual understanding of what you're going to be doing. You're going to be using a transfer pipette to move 1 milliliter of wash buffer onto your plates of worms. And so we'll do this with the wild type first. You'll add a mil of the wash buffer. And then usually what I do is I kind of swish it around a little bit to help bring some of the worms up and into the surface. And then we'll tilt those plates and then use a new transfer pipette to basically suck up the worms and the liquid that we just put onto the plate, and then move that into the transfer, or sorry, the 1.5 mil tube that was labeled wild type. We're going to be trying to conserve our micropipettes today, and this is something you'll want to do in your classroom anyway. And so we're going to be labeling that one that we use to go into the wash buffer as just WB or wash or whatever works for you on the transfer pipette. And the transfer pipette that you use to then pull the worms off of the plate, I would label that WT. So this one will touch all the wild type worm stuff going forward. So with that, I'll have you go ahead and bring that to fruition. So go ahead and take the wash buffer pipette, pull up 1 milliliter, and that's going to be the line right beneath the bulb. You're going to add that to the surface of the wild type plate that has your worms on it, and then give it a little, little bit of like, shimmy on the bench top to kind of help pull up some of those worms, and then give the plate a little bit of a tilt, and that'll help those worms kind of roll down to one corner, and that way you can kind of pull them up with your wild type pipetter and put that into the 1.5 mil tube. And for those of you with really fast hands, because there's always some of you in the group, you could move on and then execute the same process with the mutant plate. And so it's going to be the same thing. You'll take your wash buffer pipette, you'll move 1 milliliter of wash buffer to the surface of the mutant plate, give it a little shimmy, tilt it over to one side, and then pull up that liquid containing the worms and move it to your mutant 1.5 mil tube. If you've got some sort of rack around, you'd want to place your tubes now that have the worms and the wash buffer kind of in that rack to hold them upright. If you don't have that, you could just hold the microcentrifuge tubes in your hand. So the reason we want to do this is we're trying to get happen with the worms is to get them to precipitate to the bottom. And this is going to allow us to basically pull off the wash buffer that's over the top of them. Because the goal of the next couple steps is to basically produce C. elegans worms that are really clean. We're going to be literally washing all of E. coli off of them as well as we can. So that way, when we move them to our assay plate, these worms are going to be hungry and looking for food. And that's what our entire assay is based upon, is their ability to associate salt with food. And so that's why we're doing these wash steps. So those will now sit kind of upright for about 2 minutes. And like I said, if you've got a rack to put them in, great. But if you don't, just hold on to them in your hands, and that will work just as well. And of course, sometimes, you have kids where you don't want to give them the racks because they've got those type of hands. And so this is a way to keep them busy for at least 2 minutes of that class period. If you've got those worms and you're holding them, another thing you can do is you can start looking at them and you can watch them pellet. And so depending upon how good your eyesight is, you might be able to see it really easily, or you could take your cell phone light, turn it on, and then shine it at the side of your micropipette, and that will give you a higher contrast in that microcentrifuge tube, and you'll be able to see a small little white pellet developing in the bottom. And what you'll notice is that wash buffer is probably pretty cloudy. All that cloudiness are those OP50 E. coli that the worms have been feeding upon for the last couple days. And so while they've been in there feeding upon those E. coli, they've been learning to potentially associate food, those E. coli, with the presence of salt in the NGM plate. And one thing also to bring up here, because depending upon the students you have, I always joke and say it to the AP students, but it's not always them, it's just any student that's really meticulous, is not every worm's going to make it in this protocol. So don't spend the time to try and get every worm from that plate that was in the tube to the next round. there will be some casualties, and that's just it's part of life for the worms in this case. So we'll try to keep as much of that pellet as possible, but if you lose some worms along the way, don't sweat it. Really, at the end of this, we really only kind of need 10 microliters worth of worms, and so you will probably have plenty of worms there as you finish this washing protocol that we're going to be running. All right, that's about 2 minutes at this point. And so most of you, if you check, you should see that you've aggregated worms or a white cloudy blob down to the bottom of those 1.5 mil tubes. And what I'll have you do now is using the appropriate transfer pipette. So you've got the pipette that's labeled wild type and you've got the one that's labeled mutant. I'll have you remove as much of that buffer as you can without disturbing the pellet. And so what I would do is I would kind of squeeze my transfer pipette first, go in there, and I like to put my transfer pipette just beneath the meniscus and then slowly release my thumb and suck it up and then just kind of move my pipette down as that meniscus moves down. I feel like that gives me the least amount of turbulence. And I'll just kind of get down close to the pellets. I won't go all the way down to the pellet. And then I'll call that good enough. And then I'll take that waste and I'll squirt that in my waste container cup that I have at my bench. That waste container, and this is things to think about with all these materials, is that's containing that OP150. And so this is ultimately going to be something I probably want to bleach if I'm just doing this in my house, or if I'm someplace where I've got an autoclave, I will then be bagging this up and putting it in the autoclave. But I'll have you go ahead and finish up pulling off the wash buffer from your wild types and from your mutants. And
Damon, if you would clarify, should they disrupt the pellet while they're siphoning off the waste?
Yep.
Should they let it settle, or should they continue to progress and just take what's remaining of the pellet?
You can just take what's remaining of the pellets. Like I said before, we're going to have so many worms at the end of this wash compared to the number that we actually need to put on the plate at the end, that if you're losing a portion of them due to a slightly unsettled pellet, it's going to be okay. Thanks, Tamika, for grabbing that. So now that we've kind of washed them once, they still have some E. coli around them, right? There was some cloudiness down that pellet. And so we're going to do a subsequent wash. So using that wash transfer pipette, we're going to add 500 microliters to both the wild type and the mutant 1.5 mil tube. And so since we're going to be using that same pipette both times here, I would say it's really important to make sure that when you're adding the wash buffer, don't go all the way down inside the tube with the worms. Just go ahead and open your 1.5 mil tube and kind of drop the wash buffer from the top, and just do that for both of them. And as you can see in Lee's, the ones there, what you notice is it's not quite as cloudy as last time. So we've removed a fair amount of E. coli from that first wash. But remember, we're trying to get these as clean as possible because that means they're going to be as hungry as possible when we put them on the assay plates. And so what I would do at this point is take them and invert them kind of just gently 5 times. And what that's going to do is going to create enough turbulence to pull any E. coli that is coated on the worms off of them. And that means we'll be able to get a little bit slightly cleaner of a pellet.
That was a good tip. Mary said that she has her students save the washes, so they could absolutely put them into a separate tube in case they accidentally create a worm pool. This is I like the worm pool. So that's what happens when you depress the bulb of the pipette while it's in your worms. They kind of go for a little spa, but they can also just let them sink down to the bottom again if they do that. So it only takes a minute or two.
But to Mary's point there, if they've got a group that accidentally, because remember, some students with transfer pipettes, weird things happen. If they accidentally suck up all of the worms and then go and put them in the waste, if you were just to give your students an additional set of 2 mil tubes for each station, they could then put basically waste just from one worm type into each one of those tubes. So if something did come up, they would still have their worms in there. Or the other thing to think about here is you still have a ton of worms back on those plates. And so if you do have a critical blowout where all your worms aren't in the worm pool anymore and aren't in the tube, you could have them start back over at the plate and rewash the plate again and start from the beginning. So those are two solutions for that issue. Once you've given them that nice little mix, we're gonna do the same thing as we did last time, which is set them up and then let them settle for approximately 2 minutes. And this is one of those protocols, right, where we're looking for a specific outcome, which is just worms pelleting down at the bottom. And so that could happen in under 2 minutes. It could take longer. So there's always one where students should be doing a visual check to be like, yep, we've hit the desired outcome, and now I can move on to my next step. So usually we would do, or at least when I do this one, I usually do at least 3 washes because I like the worms to be really hungry because that way I know I'm getting better results usually. And so I think, do we have enough buffer to do it 3 times?
We have about 10 mils of wash buffer.
Oh yeah,
We
you guys
have
are good.
plenty.
Yeah,
Yes.
plenty. Okay. Yeah, wash buffer is something you don't necessarily need to be stingy with when you run this kit. And so that's always something to also keep in the back of your mind. You're not running PCR or something like that. We were like, oh, I don't want to waste any of that reagent. Go ahead and do it to make sure that the worms are nice and clean. So that's been about 2 minutes from when we did the last pelleting. So go ahead and repeat what we did before, which is take the appropriate pipette. So the wild type pipette for the wild type 1.5 mil tube, and pull off the supernate from above the pellet and put it into your waste container. And then do the same thing for the mutant worms as well. And Lee, I have to say, I actually really like that 1.5 mil rack you have. I like the size of that.
This is so handy. I know. I Simport.com, I think. Yeah, it is super. It's good for webinars. I can't usually fit the big green one on here.
Yep. And what I'm usually using for worms is I will use the floats from Pavlov because that gives you enough holding space to be able to kind of perform this lab. And what you can do is you can basically give them all the reagents they need to run this lab, the wash buffer, the 1.5 mil tubes in that. And so that way you can just drop it off, and that's kind of their lab station with some pipettes.
Know why my phone is so jumpy today. It's not usually like that. It's the focus. I think it's having to go back and forth, but
it had that afternoon coffee, I think.
Yeah, right. You can
so,
definitely see that mine's getting a lot clearer now.
Yep.
I'm gonna let them settle.
Once you've got that pellet all the way down again, go ahead and add our next series of washes to them. So another 500 microliters of wash to your wild type and a 500 microliters to your mutant. And then once again, follow both of those up with just some gentle agitation, 5 or so back and forths of the tube, just to kind of help strip off any of those E. coli that are on the outside of the C. elegans. And then set them up again and let those little worms precipitate. And what you should see is like what Lee has seen with hers, which it's now pretty darn close to what the wash buffer looked like. And once you kind of hit that point, you know what, these worms are pretty darn clean, and I probably don't no longer need to do an additional wash. And this is usually 3 times, like 90% of the time I've run this lab. I've only had like a handful of times where I've run it where I don't know what happened. My E. coli culture just went crazy, and it took me 4 washes in order to get them clean. But most of the time, by the 3rd one, you're going to be super, super clean, and you'll be able to move on to the next step. So I'll just kind of let the timer go down for another minute here while your worms make their way down to the bottom of the tubes.
I'll also mention, because I'm noticing it on mine, that I have a lot more mutants than wild types. And that was true when I looked at my plates too. I don't know if you guys can see the difference in the size of the pellets. And that's okay. We're actually going to do a little ratio of worms. So we're not necessarily comparing the, one type to the other in terms of number. So don't worry if your worms are more crowded or more uh, if there's more worms on one plate. I'll let Damon talk about crowding later on though.
Well, this is something that I see usually every time I run this is I rarely will have mutant and wild type with the exact same kind of proportions when I'm doing the washes. And I think this is just depending upon those starting conditions of the worms getting on the original plates and kind of growing out, they'll just end up at different kind of overall proportions on the plate. So like Lee said, it's nothing to worry about because we're going to be sampling such a small amount of them and then just doing an analysis on a small statistical portion of them. So right there, we're at about the 2-minute point. And so you can remove the excess buffer if you would like at this point. So that way you've just got the pellet down in the bottom.
I've forgotten what wash we're on. Is this 2 or 3?
That was 3.
Okay.
Yep. So you're nice. I can even see it on the little tiny pixel screen I have of your hands. That looks pretty darn clean. So I think
Yeah, it looks
we're
clean.
good at
Yep.
3. And so this step is one I have some teachers do and not do, which is remove the buffer on the last wash, because ultimately we're just going to go down and pull worms off the bottom. And so if you're somebody who loves it just to cut out steps, you could cut out and just go to the bottom. But the caveat is I found it with students, you need ones that are kind of like high performers, because otherwise what happens a lot of times they'll make the worm pool happen and then you'll get a dilution of worms that end up on the plate. So I think it is much better to do what Bio-Rad's protocol has, which is remove that last volume of wash buffer, leaving behind just that pellet in the bottom of each one of those tubes. Traditionally, once you have those worms in the bottom of the tubes, you would have the students use a modified P20 pipette tip in order to be able to move the worms onto your assay plates. And the way you would do this is you would take a pair of scissors and you would clip the tip maybe about 20% up from the bottom. And the reason you're doing this is so that it makes a larger borehole in the pipette tip. Because if you go to the really small borehole in the pipette tip, when you bring up the liquid with the worms in it, it creates an extreme amount of shear force. And what you end up with is a worm smoothie. And it turns out when you put a worm smoothie on the plates, they don't go anywhere. They're all dead. And so that's why you've got to cut the pipette tip if you're going to be using pipettes for this. But you also don't necessarily have to use a pipette tip to do this. It's nice because the pipette, right, gives you really, really tight control over the volume. So you can be like, I know I added 10 microliters of both my pellets there. So what we're going to do today is you're just going to use your transfer pipette and you'll go down into the pellet and then just take up the smallest amount you can, basically just try to get a drop. And then you're going to bring that over to the appropriate assay plate. So like my wild type worms are going to go onto my wild type plate and I'm going to put them square in the center of the plate. So you broke the plate last night with a line down the center. You're going to put it right in the center of that line and just one little drop from each of those worm tubes should go to your assay plate. So I'll have you go ahead and do that now. Sorry, I'm going quiet because I'm watching Lee's hands. um, one thing to also think about with this lab is what your experience is kind of the final activity or the final lab that the students are doing. And so they are actually working with these worms kind of previous to this. So they're doing the subculturing of them, which Lee and Tamika did. And they're also setting up the plates the day before and things like that. And so the students do get a lot of practice with the worms before moving to this kind of assay stuff that we're doing right now. And one thing you can kind of build into that as they're working with the worms previous to this is have them throw underneath a microscope if you've got dissecting scopes around, so that way they can kind of get familiar with what the worms look like. If you really don't like a student, you could have them look for male worms, which are like 0.05% of the population usually.
I've yet to see a male that I'm aware of, at least.
I saw one years ago at Rock
I
Canyon High
don't believe
School
you.
in Colorado. I was very, very surprised. But we'll show you a diagram of what a male looks like a little later on here. But now that your worms are dropped onto the plate, what you should see is you've got a little bit of liquid there. Some of that liquid will probably get absorbed into your plates. And then the worms will be able to kind of start making decisions based upon the environment around them. So one side of the plate, you had a drop of sodium chloride. And on the other side of the plate, you just had a drop of your wash buffer. And so they will be making these decisions of where to go based upon looking for food. If you've got a really, really big puddle of wash buffer with your worms, it's going to take the worms a while to kind of hit the ground running, get to the auger and move. And so what you can do is you can take a paper towel or a Kim wipe, and you can basically just make a little tiny tip with it. So just kind of wad it up into a little tip. And you can take this tip and then just touch that drip of water, that drop of wash buffer, and it will suck up the wash buffer, and that will allow the worms to get in contact with the medium much, much quicker. And that will basically buy you back usually about 4 to 5 minutes on this lab. And so know that that is a trick to be aware of. And of course, that after it's touched there also goes into the waste container that you would bleach or autoclave. So
So I don't know if you can see this, but see how the worms are kind of trapped in that little bubble? Is that working up there?
yeah, I can see it on my side. Yeah.
Okay. And so I'm going to, now you can kind of see how they're doing the Harlem Shake in there. I'm going to do the Kim wipe and just kind of roll it up and suck up some of the liquid, and you'll see how they'll start crawling out of that bubble before. So give me just a second and I'll do that.
In the interim, Damon, we have
Yeah.
a question in the chat.
Yep.
I know that theory is coming later. Don't know if you want to address this right now. Nikki Mertz asks, which wild type of mutant strains are you using?
Yep. We'll get to that in just a couple minutes here. We'll have Lee show this tip, and then we'll jump into kind of the theory behind this lab, which is going to dig into the mutant type, and then also kind of the neurology of the mutants we're using.
Okay, and we have one more question that just popped up as you were
Yep.
addressing that question. Why did you say the pipette tips
Ah,
need to be cut?
so they need to be cut because if you use a standard kind of P20 tip, it's so narrow that you will get a shear force when you try to pull up the worms, and it'll cause the worms to actually be ripped apart. And so all the worms will be dead if you use a standard pipette tip. And so that's why you want to cut the tip. That's also why, to some degree, I kind of prefer the transfer pipettes, because if the kids have gotten good with them, I don't have to worry about that. I don't worry, about, oh, we forgot to modify the tips. We've sheared them half of our worms. So that's the reason you're using the micro or the this transfer pipettes today with this lab. All right, Lee, it looks like yours now have contact with the media and are running.
Yep, they're going. They're worming.
Yep.
Okay, I'm going to put my video off and I'll come back to this in a little bit.
Okay, sounds good. And so you can take your plates now. You can just kind of set them to the side. What's going to be happening now is your worms are going to be making those decisions. One caveat, though, because I have run into this in one course before, is if you are in a room where you've got bright light, you want to at least orient the plates so they are the same distance from the bright light, because these worms, right, they're not hanging out on the surface of the soil. Usually they are kind of down below, and they're not a big fan of super bright light. And so if you've got a super bright light around, you might get a false positive, false negative, because they're trying to avoid, bright sunshine. So just be aware of that.
And Damon, we have a very, I guess,
Oh, yep.
kind of measured question here. Do we put the plates upside down or right side up?
Yep. Go ahead and keep them right side up in this case. It'll just make it easier for us to kind of visualize them. And usually the reason you invert plates when we do bacterial plates is because we're going to be growing them and we worry about humidity building up and causing drops. They're going to fall down. But we're going to assay these plates at the end of the webinar, and your students will do it, in 20 or 30 minutes. And so you don't have to worry about humidity building up to the point where it's going to fall back down onto the plate. Good question.
I will say, right now I have flipped my plate over. Sorry, let me get this
Mhm.
where you can see it actually. And let me see if I've got this spotlighted. Hold on, give me 1 second.
It's okay. Well, Lee, while you're pulling that together, we
Yep.
have yet another question, Damon.
Okay, yeah.
So if we have dissecting scopes available, should we not use any lights on them?
So I wouldn't set up my assay and then have them go use it on a dissecting scope, because that bright light will interfere with the assay, because depending on how much time each one of those plates get, right, you have added this new variable to it. But if you just want to look at your subculture plates on the dissecting scope, go for it. Totally fine. To bother the worms a little bit is totally cool at that point. It's just during the assay, we're really trying to keep all the variables the same, as much as possible.
Okay, so this is
All right.
my mutant plate right now. I think you can see how they're just going to town. And this is again flipped upside down. So you just kind of have to adjust the focal plane of your camera um, or your microscope. I will show you, I've got a macro lens. Oh, hold on. I've got to show you this way. There's a macro lens on this. We have some links. I think that I sent you in the prep videos, but I mean, really, they're not very expensive on Amazon, and they work really well. I will say that the iPhone 14, if you have a 14 Pro, you don't need the macro lens. It really doesn't do that much. But you can see it works really great with
Yep.
the older iPhones. So
And these
yeah.
macro lenses that Lee's talking about, like she said, they're relatively inexpensive. I used to be able to get them for 50 cents a piece on Amazon, but now I can usually find them at least under $5 for like the cheapest ones, which are the ones I buy. Um, and they basically replace, a very budget in dissecting scope. This can get you 20 to 60x magnification, and it just fits over the top of the phones of the kids, which is also nice
Oh my gosh.
because the kids will take pictures with their phones, and now you've bought more brain space because you're on their brain device, and they'll see pictures of the worms later and show other people them and stuff like that.
So
Okay,
Damon, I don't
Damon, we
want
have
to
a
give
couple
away the.
things in the chat. Sorry, Lee.
Go ahead.
One, could you please explain the paper towel Kim White trick
Yep.
again and or show it? We'll start there.
So we demonstrate that previous there. What's going on is when you transfer those worms, sometimes you have a pool of wash buffer that are still all over the worms, which makes it hard for them to get contact with the NG or with your assay plate. And so a little chem wipe or paper towel, and if you go in there and touch that drop with it, you can suck up the liquid into your paper towel or your chem wipe, and that then gives the worms quick access down to the plate. It just makes it so the assay takes faster, because the worms won't have to swim out of the wash buffer in order to kind of start exploring.
And
And then,
people usually ask if you lose some worms in that. Yeah, you do.
Yep.
So if you have a situation where your students have subcultured the worms, we subcultured for you guys, but they'll subculture as a lab activity. If you have a group that has subcultured worms and they don't have much of a worm pellet at all, maybe don't have them suck away the extra liquid. You want to make sure you have enough worms to see. But in this case, we have gobs of worms.
And Amanda brings up a really good comment in the chat, which is worms are temperature sensitive. And so you don't want to expose them to extreme amounts of heat, and especially some of those older scopes with like hot light bulbs. You can't get to a point where you could kill worms if you had them underneath there for too long. Also, worms are not E. coli. They do not like to be at our body temperature of 37 degrees. And so the temperature worms really prefer is just ambient room temperature in most places, unless you're someplace excruciatingly hot. So their sweet spot is closer to 30 degrees centigrade.
Mhm,
So just keep that in mind as you are raising your worms.
mhm. So I think
Um,
that you somewhat address Kristen's question, Damon. How can we preserve the worms that we're not using? If so, how can we store them properly for long periods of time? And that might be part of the conceptual pieces that you'll be addressing here shortly.
Yeah, I'll just handle that one really quick. So worms, the easiest way to actually store them is just to keep them at the room temperature, let them grow up, and then subculture them every couple months. That's what most researchers would do. Some people will freeze them, but you have to make sure you've got enough glycerol. You've got to freeze them down at -80. So that becomes something that most high schools aren't doing. But I do have people that will keep their worms year to year by just subculturing them. So they'll just pour extra plates, and then every couple months, just take some of that agar and move it to another plate and then let them regrow.
So Damon, I don't want to give away the punchline here, but I wanted to just point out that this is my wild type plate, and my salt dot is on this side. Let's see if I can get the right. I mirrored image, so I'm trying to like figure out. Okay, here we go. Right, and then my buffer side, my control, is on this side. Okay, so you can just kind of keep that in mind for later.
Yep. And we'll probably be able to see that within 10, 20 minutes. You'll have a really obvious sign of what decisions your wild type and your mutant are making. So in that interim, now we're going to back up and do the theory that you would usually do in your class before starting all of this lab stuff. But we just wanted to make sure that we had enough time that the worms would assay for you while we were all here. So C. elegans, like we briefly touched on in the beginning, is a model organism. Why do we use model organisms? Well, it's a degree of bioethics and a degree of things that are just easier to work with. Humans are pretty hard to work with a lot of times, as many of you know, being teachers. And a lot of things that we would like to study in a human, we just ethically can't. And so a model organism is preferable in many, many ways. And so there are a variety of model organisms out there, and a lot of them are chosen based upon certain attributes of them that allow us to study things that are going on in a human and give us the ability to do that at kind of like, a price point that works, a time scale that works, and an ethical structure that works. So if we think about the different model organisms that maybe some of you are aware of, we can kind of talk to what some of the discoveries have been in them. So yeast, classic one, right? We've been growing yeast for, since at least 6000 BCE is the oldest protocol I found about growing yeast. It was basically making wine in Armenia. So, this is an organism we've been very accustomed to growing. We're really good at it. And we've done a lot of great biology in it. And so yeast is really what allowed us to understand cell cycles and DNA repair. C. elegans worm has been amazing because it just hasn't been around that long. I mean, Sidney Brenner brought this in just a couple decades ago. And we've learned so much in this system. So we've learned about programmed cell death. We've learned about aging, brain circuits, and it's just been a treasure trove, I would say, of useful discoveries because it's such an easy organism to work with, because the size, it's easy to upkeep, you can see right through it, which made a lot of like the programmed cell death and even kind of developmental biology stuff easy to do, because you could basically drop GFP into cells and then see where they migrated, see what was going on, because you could just do that with a microscope and UV light. The fruit fly, of course, has been a model organism since, Thomas Hunter. So we've got innate immunity was really kind of flushed out in there. It's still used as a neurodegeneration model, although I think C. elegans is creeping in on that territory with the fruit fly. And sleep disorders are also well studied through fruit fly models. And then the mouse, of course, is one of these classic model systems that people think about all the time when we say model organism that's going to give us something, hopefully, that is very, very similar to the human. And so, a lot of gene expression work has been done in mice, epigenetic work has been done in mice, cell signaling, and then diseases like cancer and cystic fibrosis. And what you'll notice is, the closer you get to the human, the closer we get to kind of nuanced things that we think about just happening in humans. And so that's why we're using, a rodent model in that case. One other way to think about our model organisms is actually from an evolutionary point of view. So if we think about where they diverge from each other, it can kind of give us a general idea of, maybe how much of the cellular activity in that organism might model back up to the human. And then, not too surprisingly, kind of the further distant organisms from us tend to be easier to work with, just because they tend to be smaller and easy to upkeep. And so kind of, at the bottom of the eukaryotes, we usually tend to think about C. elegans, or actually one level beneath that, I guess, would be yeast. But then we kind of have these other ones that scale up towards the human: Drosophila, zebrafish, xenopus, chicken, and mice. Not represented in this is even the bigger evolutionary diversion, which is going back and looking at things like E. coli or some of the archaea. But we do a little bit of work in E. coli that then scales back to humans. But if you're really talking about doing drug discovery or disease stuff, you need to be in that multicellular organism. And so getting as close to human as you can evolutionarily makes sense. So worms, the reason that they have kind of taken off is they're just so easy to work with. They've got that small size. And so, I mean, you literally could run your lab with a couple of desk drawers where you could just hide your worms. They've got a really rapid generation time. And this is a huge thing when it comes to a model organism, especially if you got, grants and things like that. You want something where you can fire away new experiments day in and day out. And if you've got an organism where you've got to let it grow for 2 or 3 weeks, or maybe months, if it's a really large eukaryote, this is eating through everyone's time. They're also super easy to upkeep because if you can basically grow E. coli, you can grow C. elegans. The experiments are usually pretty easy to set up with them, very similar to like what we're doing here with just this Chemotaxis assay. And then the other big benefit, and this is mainly on the backs of researchers in the last 20 to 30 years, there's just this high volume of existing information. And the worm community is, they're really wild because they're so passionate about their model organism. So you can go to Wormbase and kind of see all the things going on in the worm community, where they freely are trading a lot of the mutants they like to work with and techniques and things like that. So they're just a really cool organism to work with. They're also really safe because they don't really have any sort of pathology with humans, but there is still a lot of genetic homology to humans. Nobel Prizes that have been basically won on the backs of worms. We had physiology and medicine, 2002. Sidney Brenner, the gentleman who brought kind of worms to the forefront, is in there. And they basically kind of laid the foundation on how genes control the fundamental processes of animal development and cell death. And so they're ones that basically figured out, once I got my fertilized egg and it divides, where do those 2 cells go? And then when they divide again, where do those 4 cells go? And how do I basically follow this chain until I have the entire C. elegans body of 939 cells? In 2006, Mello and Fire won a Nobel Prize for basically discovering RNAi, so interference RNA, this RNA that can go in there and basically modulate cellular activities of organisms. And this is one of those ones that kind of blew me away because when I graduated in the early 2000s and I had people that were asking me questions about this soon after I graduated, and I was like, I don't even know what you're talking about. Because it was one of those things that really just kind of came out of left field. We didn't have any kind of even inclination that this was taking place until kind of the early 2000s. And then also chemistry wise, there was the use of GFP within C. elegans that Chalfie won a Nobel Prize for in 2008. If we think about where we can find C. elegans, they're basically distributed worldwide in kind of the temperate zones. So most of North America, you would be able to find C. elegans in any sort of kind of moist soil area. They really like areas around kind of riparian habitats or compost, basically any place where you've got a lot of rotting vegetation or decaying matter. That's where those C. elegans will be because they're there to eat the bacteria. They're non-pathogenic, like we said again, so that's also a really, really big benefit about using them. We talked about how easy they are to culture in the laboratory, and you have some experience with that under your belt now, since you've had them in your hands for a few days potentially, and you could try to hold on to your plates for a while and see how long that plate you have of starter worms goes. And what you'll notice is as long as you keep them out of the heat, kind of keep them from getting dehydrated, they'll be able to go easily for months upon that plate before they kind of crowd it out. And then once they've crowded it out, then they start to kind of decline. And one thing to consider there is just remember they don't like those warm temperatures. So being, 20 to 25 C is probably the best location for them. And then as far as reproduction goes, they reproduce about every 3 to 5 days. As far as the division of sexes per se in the C. elegans, the majority of the worms you have are hermaphrodites. So those hermaphrodites can lay upwards of 400 eggs. This which gives you that really, really quick regeneration. And then males, as we joked about before, are a really, really small portion of the population because they're just not that important. So 0.05% of a C. elegans population is usually males. And the way you can tell them is underneath the microscope, the males will have kind of this fan tail at the end. And like Lee said, like she's never seen one. I've only seen one when I was visiting a high school and they were running this. So they are kind of like an Easter
They're
egg hunt
mythical.
to find. Yeah, they're magical. They're basically unicorns. The life cycle of C. elegans, you will go from your adult through 4 larval stages. And so that hermaphroditic adult can produce an egg. That egg then will produce your L1, and then L1 can go kind of 2 directions based upon the environment. So if the environment is unfavorable, so it's bad on temp, it's bad on food, it'll go into this dour state where it can kind of pause itself out. Um, and then that dour state can be recovered and jump right to kind of the larval 4 stage when things get better. But if things are already good in the environment, you'll basically pass through L1 to L2 to L3 to L4 and be back at that adult within just, 3 or 5 days or so. And so this is what allows them to kind of be regenerated very, very quickly. The amazing thing too is that those eggs are really hardy, is from what I've noticed. I've microwaved my worms before in order to try and pause them so they're easier to count. And I thought that would kill them. And I left the plate out for a while. And then I realized I had worms in a couple days. And so I did it again and then watched. And what I realized was that the hermaphrodites that had eggs in them, the microwaving didn't kill the eggs, and the eggs then could hatch days later. And so I've seen this too. It's just other applications of heat. As far as their physiology goes, a lot of it maps back to humans, and this is also one of the reasons people like to work with them. So they've got an entire kind of muscular system that we can kind of make inferences from things we learn in them back to humans. They, of course, got this gastrointestinal tract, which you can like watch the whole thing. If you feed them underneath a microscope, you can see stuff move all the way through them. Or what I like to do is I like to feed them GFP, pea glow bacteria, and then shine a black light on them underneath the scope, and you can see the GFP basically travel through their GI tract. They've got a nervous system, and this is where really I'd say a lot of the crucial work for connectomes, understanding how neurons wire together to then give us things like learning, stuff like that, are taking place. And then they have a cuticle, which is pseudo-analogous to our skin.
So Damon, just speaking about nutrient requirements, Adrian asked, can NGM be substituted for another type of agar?
That's a good question. I personally haven't played around with that much, so I don't have a good answer for you. If you're just trying to do maintenance of the worms, you could probably get away with like an LB auger for a little bit, but the worms they do need a little bit of cholesterol. And that's because they've got neurons. And so you need that cholesterol in order to kind of maintain some of those neurons. And so you could probably culture them for a while on LB, but then at some point they will probably start to not do well because they're not getting enough cholesterol in their diet.
And if you can maintain that track of thought, can other types of E. coli be used?
Yes, yeah. We use OP1, OP50 because of the way that they grow. They're one of the preferred E. coli to use for C. elegans because they don't create as thick of colonies, so it's easier to see the worms in them. So that's half the reason to use the OP50s instead of, say, HB101 or something like that. If we think about comparing the worm to the human, we basically have the same number of genes. Sorry, Lee, I edited your slide just because I went back and I was like, I know that number just dropped like this year again. And yeah, we're
That
down
still
to,
blows my mind though.
yeah,
It's crazy.
well, and as a human, I'm just going to put it out there. It's not about the number of genes. It's about how you use them.
It's about how you use them.
Yeah. It's about all that rearrangement we do after the fact. That's important. If we think about the size, though, from a cellular point of view, C. elegans is only 959 cells, where we're about 100 trillion. So finally, we're beating them. Neurons, they've got about 302, which is incredible. That's almost 30% of their body, or 30% of their cells are their brain. We've got 100 billion compared to our 100 trillion cells. So we're kind of like dummies on just a mass ratio. And then chemical synapses, they've got about 5,000, where we've got about 1 quadrillion. So we're kind of beating them on that ratio. Associative learning. this is the core concept that we're trying to get through to the students on this lab, or at least that's what the assay is going after. So like I said before, almost 30% of their entire cellular makeup is neurons. We know how all of those neurons interact with each other and touch each other. Actually, caveat, we know how they touch each other, and we are learning about how they interact with each other. So those chemical interactions is what's going to allow us to understand really complex behaviors like learning, like memory, things like that. And this is definitely the organism we will figure these out in before any other. And this is really important because, what was it, 7 years ago, this project was launched, the Human Connectome, which is basically kind of an audacious project saying that we were going to figure out how all of the human neurons wire together, and then ultimately how that wiring and firing ultimately produces things like consciousness and knowledge and memory and personality and all that type of stuff. And so we are slowly making our way there, mainly in the worm first, and then secondly in the human as we transfer techniques from the worm to the human. Similarities in our nervous system is the worm has sensory neurons, just like we do. They've got interneurons. These are the neurons that kind of take that sensory information and then recode it to then make sense more or less out of it, and then kick that out to then do something about it, i.e. the motor neurons, which then is triggering your muscles to do stuff. Associated learning is something we can evaluate in worms. They are an organism you can do associative learning with. One of the organisms your other students are probably familiar with, though, is Pavlov's dogs, in this case of associative learning behavior, which is the dog would have a bell rung, it would be fed, and then what you could do is you could ring that bell and the dog would start salivating, knowing that food was coming. You can kind of do the same thing with worms, but instead of a steak, they prefer E. coli. And so what we've done is we've trained them by giving them a little bit of salt in the plate and having E. coli there. And so now they can learn to associate the presence of salt with the presence of a tasty butt bug, E. coli. So what we can do with your plates is basically put salt on one side, wash on the other side, and see, do they still have this associated learning behavior? And that's what's going on in your assay plates. So the wild type ones, right? Are they going to be able to associate the presence of salt with food? And is a similar thing going to happen with your mutants? At this point, you guys could probably actually check your plates and see what's going on. So why don't you go ahead and do that now? See if you can tell how many worms have kind of moved towards the salt or towards the wash for each of your plates. And what you'll probably see is that they have started to make choices and your plates might look a little different. So let's talk about that mutation then, so that you understand why they might be behaving differently between those two plates. So the mutation that you're working with is a mutation in DAF18, which is a phosphatase that's involved in a signaling cascade that basically takes an outside signal and then translates that into induction in these neurons. And so what happens is if you've got a mutation in DAF18, you basically can't respond to outside signals and be able to do anything with them. And what this does is it causes a deformity in the neuronal pathways of the C. elegans. So DAF18 is expressed, or you kind of see its expression strongly in the neurons here in green. And so we've got a sensory neuron and your soluble attractant neuron that basically is expressing it. And then you've got an interneuron that's also expressing DAF18. So if you're a mutant, your DAF18 isn't working in both of those. And so you can't recognize salt per se with that sensory neuron. And then even if you did, when that came into kind of the brain function of making sense of it, that portion is also broken. And so our mutants basically aren't able to detect or process information about sodium chloride in the plates. And so that's why if you look at your plates, you've probably noticed that on your wild type, all of your worms have moved over to the salt side, and probably not very many are hanging out by the wash buffer, where on your mutant plate, it might be a more uniform spread of worms on both sides.
Damon, do you want me to show my plates?
Yeah, go ahead and show your plates.
Okay, let's see. I've got to get the video on and let me pin it.
Yep.
And while Lee is pulling that up, if anyone has any questions while observing your plates, now would be a great time.
there was, okay, let me see if I can get this over. So you'll probably have to excuse my messy desk here. Now you get to see the real issue here. So, okay, the, oops, assay plates are here. I've just got them on another phone, so it's nice and dark.
Dark.
A dark background is really best. So here's the mutants, and you can kind of see they are over, I would say there's a lot of mutants on here for one. They're over towards the salt more, but there's definitely a good bit on the control side as well.
Mm-hmm.
And my wild types, it's going to be hard to, let me see if I can zoom in. They are almost all, I don't see any on the control side. They are all over here bunched up on the salt. So it's really hard to see right here. Let me see if I can see the worm. Sometimes it helps to look at the worm trails too. You can kind of see they leave this little, it's going to be hard to show in the video.
Yeah.
If you tilt it towards the light, you can kind of see where they've been crawling around. You know, they leave a little trail, almost like a snail trail, right? And so you can kind of see where the trails are on one side or the other.
And this is another point where if you're having a hard time seeing them, I would turn your cell phone light on and put it at the side of the plate, and that'll give you a higher contrast against those worms. And so you'll be able to kind of get an idea of where they're at a little bit easier using that. So your students, what they would do at this point is they would go through with a Sharpie and then mark the number of worms they're seeing on both sides. And the reason you're doing this is going to allow them to actually make a chemotaxis index, which is going to be a pretty simple calculation. It's basically the number of worms on the side of the sodium chloride minus the number of worms on the control side divided by all of the worms on the sodium chloride side and the worms on the control side. And so what you're looking for them to be able to generate is basically, is there a positive chemotaxis, or is there a negative chemotaxis, or is there no chemotaxis? And typically what you'll see with this lab is the wild types will give you a positive chemotaxis, so a value above 0 and below 1. And the mutant worms will usually give you close to no chemotaxis, so kind of like 0.2 to 0 to minus 0.2 or so. And so it can sometimes, I guess that's the end of the slides. Sometimes it's hard for them to be able to visualize them quickly enough. And so I would say for most of them, I say, just focus on counting them as fast as you can within 1 minute. And the best way to do this is with a Sharpie pen, and have them hold it up to the light and then just dot really quickly where they see the worms. And this will allow them to kind of get the values they need in order to run that index with. One of the slides that I just realized didn't get shown, I think it was accidentally hidden, was to show the homology of what DAF18 is in a human. So DAF18 is very similar in its pathway to a gene that we see with autism. And then also the receptor that's in the pathway of the worm, where you have the signal coming from the outside, DAF18 is acting on it in the signaling cascade. The receptor of that system, if we look at that receptor within humans, we find that associated with diabetes. And so this pathway that they're working on has kind of direct correlations to known human diseases, but it's actually still a pathway that we're still having a hard time working on, because ultimately a transcription factor gets initiated throughout this pathway where DAF18 is involved, and we still don't know all the outcomes of that transcription factor. So that's still a place of active study right now. But this is the way it is with, most science like this. It's like, you know a lot, but you also know that you don't know a lot, and there's more to be done. So that more or less is this lab in a nutshell. Does anybody have any questions that we could answer while we're all here?
So thank you. Clarified a couple of the questions that were being uploaded to the chat, Damon. There is one by Mary. What other stimuli can we use to induce associative learning in these worms, i.e. for student design projects?
Mary, I'm glad somebody planted you in the audience for this. You can use, what I like about this is you can basically open it up and have students find ways to go about this, because worms will respond to lots of stuff. So let's talk about some of the stuff that you could accidentally do that might get you different results. And that would be if your worms grow for too long, what can happen is they can start to associate salt with living in a busy, crowded place they don't like. And so when you go and you do the assay, what you'll notice is that the worms basically go away from the salt. The wild type worms go away from the salt because they learn to associate salt with being crowded, and that's not something that they like. Or say if you grew those plates at a high temperature, so like, above 30 degrees centigrade, they will start to associate salt with warm temperatures that they do not like, and they will actually run away from the salt in the assay. You could do a similar thing with sunlight. I've seen a couple schools try things like caffeine with them, other types of stimuli, chemical stimuli, to then see if they can get a positive or negative chemotaxis from it. So the beauty about having them do this lab, I think, is it could set you up for having them now design their own experiments based upon the scaffolding from this lab, because they can run a very similar model, which is add this thing so the worm associates food with this thing, and then assay that later to see, does that association hold true? Is it a positive association or is it a negative association? Lee's brought up the slide that lets you know how much this kit services. So traditionally we say that this lab will serve 32 students, which is 8 lab groups of 4 people. And this is actually one where there's enough plates moving and things like that, that I think 4 students is appropriate. There's enough for everybody to kind of work on. There's the 3 lab activities we talked about, the culturing, the subculturing, and then the assay. You end up with all the materials you need to run this lab in the box. And what I like about this one is you really don't need, per se, any other hardware. Like we showed you how to do this without a micropipette. You don't necessarily need an incubator. You could just have a desk drawer someplace to do this in. The one thing that's very different, though, about this lab compared to other biorad labs is we don't send you everything at once. We send you all of the stuff to prep your plates, make all the student stations, but we keep the worms cold on our side until you say, hey, I'm ready for them. And by ready for them, we mean you've poured your plates, you've got E. coli growing, and so you're ready now for worms to show up and for you to take them out of the cold storage, basically, and put them onto plates with E. coli so that they're ready to feed. And so there'll be a little kind of call-in card that comes with the box. So that way, when you're ready, you basically call in and say, hey, I'm ready for my worms, send them. Katie, you just asked, could you please repeat how the initial association of salt and food was made for the worms? Yeah. So the plates the worms are growing on have salt in them. And as they're growing there, they are consuming E. coli and they're having that sensation of salt around. And so that's how we're getting that associative learning behavior to take place.
The other thing I was going to mention about the worms was when you get them and thaw them, do not hold them in your hand to warm them up. Just let them sit on, just let them sit on the desk. It'll take 10 or 15 minutes. That really slow thaw is very helpful. The other thing is that, and Mary mentioned this, so if you get your worms and you plate them, they don't like being frozen, obviously. Most of them will be dead. And the ones that survive are very small. And so it'll take a few days before you really start seeing them. If for some reason after a few days, one of your plate looks good, or your wild types look good and your mutants aren't looking good, or vice versa, or none of them are looking good, please give tech support a call and let them know, and we'll send you more worms. That, sometimes it happens, it's with the thawing or the shipping or whatever. This go round, my mutants were going bananas and my wild types were, there weren't as many of them. And that's, like I said, it's fine because you're doing that index. But that is one thing that would really help, is to not keep them warm. The other thing that I wanted to do was put up, let's see, am I still sharing? I'm sharing the, hold on, let me go back here.
While you're doing that, I'm going to answer this question that Mary and
Yeah.
had in the chat, which was, would you add the caffeine or the essential oils, whatever you're trying to associate with them, to those original plates? And yes, you'd basically kind of put them in the same position where salt was being put to see if they will do that association. I don't have a concentration of caffeine off the top of my head to share with you. And so I would just try to find some way to get a normalized concentration that's relatively low. So, definitely way lower than like a cup of coffee situation. And that'll be somewhere in the ballpark for them where they'll be able to pick it up, but it's not going to necessarily cause any major death to them.
Let's see. Oop, hold on. I'm sorry. My computer's going wonky. Where is am I sharing still? I'm sorry.
Yep. Yeah.
There it goes. Okay. So yeah, there's some little proper care and feeding of worms in this PowerPoint. They do not like being in an incubator, so please don't put them in an incubator. I showed up to a workshop where someone helpfully put all the worms in the incubator, and we had no workshop after that. So that's one thing just to be careful of. And yeah, that's all good. Regarding other experiments, two things that we talked about. One of them was Jose, was the R&D scientist who was developing this kit, and he mentioned that one of the cool things that he had done was try to put salt in the wash buffer. So when you're washing the worms, they don't like that. They're basically drowning a little bit. So if you put a little bit of salt in the wash buffer, you will see them hightail it away from the salt side, or more so, right? And so it takes a little bit longer of a wash, but that's a really cool experiment. I've done that with students, and that one's pretty neat, and have them think about why that might be happening, right?
This is stressful.
Yeah, we don't like this. The other one is worms like diacetyl stuff. And so if you take like butter extract, like the butter, like vanilla extract stuff, you can get butter flavored, that, put it on one side and put like real butter on the other side, they will tend to move towards that diacetyl flavor, right? Or that scent. So that's a really cool one too.
And it's interesting because people have used worms in really wild chemotaxis. So there was a research group in Japan that was taking urine from patients, and they found an association where the worms liked the urine from patients with cancers. And so they were using it almost as an assay to determine, I forget which cancer it was, but I was kind of like, who even came up with that? Is like, hey, this is what I want to run as my experiment. But even just this simple idea of being able to do a chemotaxis, because these are a very sensitive organism to picking up molecules, there's just a lot of ways you could take this.
The other one that's fun for students to do, and this doesn't have anything to do with chemotaxis, it doesn't have anything to do with wild type and mutants, they'll both react this way, is they're sensitive to touch. And so if you the way that they usually do like a worm poker is they take a little toothpick, so pretend like this is a toothpick, and you can pluck out like an eyebrow hair and glue it onto the bottom. So now you have like a little toothpick with your eyebrow hair kind of stick. The green screen isn't doing me any favors. Okay, like that. And then you can poke them on their nose or their tail, and they'll move away from that. So that's kind of a fun one too, if you're into worm torture, light worm torture. So it doesn't kill them. So that's kind of neat. I put the survey up, and I will put also a link in the chat, and we will send the recording and the PowerPoint to everyone. If there's any other questions, we'll be here, but I just want to put that here. Oops, I put the wrong thing in the chat. Let me go ahead and get that link back for you. Okay, here's the survey link. We would love your feedback on this webinar. And we also like to know what else you'd like to see, because we're planning the fall now. The new webinars are live. We have not sent the email. So if you want to get a jump on everyone and get registered for the new webinars, they are at the link that I'm about to put in the chat. So hold on for that. Okay, I think Zoom is so weird about the links. Okay, that worked. Okay, great. So that's where our webinar links are. Yeah, I mean, good grief. So, but yeah, let us know if you have any other questions. I'm going to go ahead and stop the recording now, but we'll stick around for a few more minutes.
Yep.
All right, thanks.